biases that get introduced into medical research, where if you didn't know better, you would treat what you could see. And this is an instance. And as that film alluded to, just targeting that pathology alone, what you could see down the microscope, we actually can get rid of it. We can get rid of the spots, but the disease continues to rage on. So something else is going on. And that's been our philosophy with the research, that it's not as simple as just the plaques or just the tangles. There's something else in there that hasn't been considered. So the work that we've done here has focused on that something else, which brings me to the next slide. So I've been at this for a long time. I actually did my PhD with Colin Masters shortly after he purified the amyloid out of the human brain. So I studied amyloid a lot. But the question always is going to be, what is it about aging that makes it a risk factor for Alzheimer's disease? And so that's where we've come from. That's where we think the best clue is. The main question that should be answered is, what is the chemical change in the brain that occurs with aging that increases the risk of Alzheimer's disease and might act in synergy with that pathology that I mentioned, the plaques and tangles? And so we've hunted for this. And how do you hunt for something like this? Well, you have to study normal people, you have to study normal animals, and you have to study the process of aging at its absolute quintessence. And that's why when we do medical research, sometimes we have to unravel the whole situation down to its really primitive primordial level. And for that reason, we think about the origin of life itself. And you may or may not know that life on Earth is a consequence of the emergence of metals into the crust of the Earth approximately 3 billion years ago that set up a kind of chemistry of self-replicating molecules that depended upon the availability of iron. And as a result, all life forms on Earth, with very few exceptions, rely upon iron in order to develop, in order to grow from birth. So life depends upon iron. This is like biology reduced to really basics. Life depends upon iron, but here's the interesting thing when you study the biology of iron. There's no pathway for excreting iron. Now that means that if you're a healthy animal or a healthy human being or a worm and you encounter lots of iron, which you think is great for your development, in humans it's needed for the development of the brain, it's needed for the development of blood and so forth. Whenever you encounter it, you gobble it up. And this is, all life forms on Earth are like this. But the problem is that because evolution has made us so hungry for iron, we have no means of getting rid of it when it becomes excessive. We just store it, we put it away somewhere and keep it for later on the assumption that all we need to do is reproduce. But of course, as far as humans are concerned, there is this great era after reproductive life where you go and do other things. You have grandchildren, you take up another vocation, you learn. There is this great epoch that is not something that evolution ever considered. But what happens is you continue to accumulate that iron. And of course, that iron eventually becomes too much in terms of its storage, the storage in the tissue, and it can actually literally rust in the tissue where it's collecting. It can form things like liver spots on your hands, or in the case of the brain, it can actually cause metallic rust to occur, and we can see this. So here's a theory that we've been working on for a while, working with basic organisms, and that is that death itself occurs at a point where there's so much iron in the body, a catastrophe occurs and the cells die because it's just full of rust. Now you can see this in little organisms. You can see it in flies, where you can measure the amount of iron in a fly as it gets older. And reliably, from its birth until its death, about 2 to 3 months later, the iron levels go up and it doesn't need this much iron. It reaches a point where it gets so saturated with iron. Now this happens in mammals too. This is some early work that we did on the levels of iron in the brain of mice as they age. And the iron levels go up, and they keep going up. And as long as the animal lives, it keeps going up. And you can see this, other people have looked at this as well. And they've found it in rats, and they've found it in humans. And they've also found that this accumulation of iron in humans, which can be measured now through using instruments like the MRI, the magnetic resonance imaging, which is used for brain scanning, that you can show that that accumulation of iron gets to a point where it's too much, and it causes an impairment. And I'm just flashing up some of the papers. To show you how we approach this in a good research institute like the Florey is that we don't guess. We try to come up with an argument, a strong argument about where would you consider making a drug. And in this case, we even studied worms. Here is a worm which has been dissected into 3 pieces. These animals are 1 millimeter long. So you can see its head, its middle, and its tail. And it's been stained with a dye for iron. And here is that same animal towards the end of its lifespan. So at 12 days, you can see the top, middle, and bottom staining brown for all the iron that's accumulated. Does it need the iron? Not at all. It doesn't use it at all. To the extent that we now have demonstrated, and this will be published later this year, that you can take these worms and treat them with an iron-trapping chemical that removes the iron. Here's the lifespan of normal worms. So to read this graph, on the y-axis, you see the percentage of living worms in this population, and on the x-axis, this side, the days. So the normal animal lives to about 20 days. But you can see how many animals stay alive at any particular day after the beginning of this experiment. By day 22, they're all dead, right? It's reached 0, right? That's the normal life curve of a worm. If you were an actuary and you were selling life insurance to worms, you would find this very useful information. So here's the trick. Can we lower the iron in this worm, in these worms, and do anything to its lifespan? And here's the amazing thing. We did indeed treat them with this iron-lowering drug that I'm going to tell you about more in a second. And we extended the lifespan of the worm so that the average lifespan doubled. This is an impressive result. These animals were actually extremely well. They had less iron in their bodies. They lived longer and they were healthier. So here's the big question then. Does this age-dependent rise in iron contribute to Alzheimer's disease? And with that, I'm going to hand over to Dr. Aiton.
So I joined Ashley's group some years back now, about 10 years, and Ashley had some very, a hypothesis, an idea that was very compelling and intuitive to me. But it was also very radical and counter-dogma. The idea that iron might be contributing to the progression of Alzheimer's disease and not these other factors that the field had decided upon decades ago were the cause of the disease. And so, given that this was such a radical idea, we faced the problem of trying to convince the rest of the field that maybe we've got another idea that's worth considering. So while iron, the potential role of iron had some very intuitive and quite reasonable, hypotheses for why iron might be contributing to the disease, we lacked the clinical evidence. The evidence that from a person who's suffering from Alzheimer's disease to say that iron is actually contributing to their symptoms. So that's what I embarked upon several years ago to try and understand, could we see the effect of iron on individuals? Now, how do we measure iron in the brain of someone? Now, you can imagine this is a very difficult thing to do. We can't, for instance, take a liver biopsy or measure iron in the blood to determine how much iron there is in the brain. But we can do this, and we've applied 3 different approaches. We can look at iron in people who have died, so we can measure iron directly in their brain post-mortem. We can also take the fluid that surrounds their brain, this is the CSF, and we can also now use new imaging technology based on MRI to measure iron in the brain. And I'll show you evidence from all those 3 approaches. But first, before I go into that, I just wanted to talk a little bit about what we now know about the natural progression of Alzheimer's disease. This has been a really, shocking discovery over the last 10 years, that we've found that Alzheimer's disease really is a slowly progressing disease. This is data that's come out of Melbourne under the work by Colin Masters. And this is the natural time frame of Alzheimer's disease. You can see here is the time where people have got Alzheimer's disease symptoms. But before they get those symptoms, there's a 20-year build-up of the pathology of Alzheimer's disease. It's a very slowly progressing disease. And so this would suggest that there are people who are walking around in the community who are quite healthy, but have this disease process occurring in their brain for up to 20 years before they get a diagnosis of the disease. And in fact, we can identify these individuals now by using PET scanning technology is actually referred to. So this is a graph of people who are healthy controlled, ordinary healthy controlled people who've got no memory complaints. These people here have got some memory complaints, we call them mild cognitive impairment, and these people have got Alzheimer's disease. And on the y-axis here we have the amount of amyloid within their brain as determined by our PET scanning. And what you can see is the really shocking result here is that around 30% of people who are over the age of 65 and are cognitively normal have amyloid in their brain equivalent to that of someone with advanced Alzheimer's disease. Yet they have no cognitive symptoms. It seems that amyloid is not alone sufficient to cause the symptoms of Alzheimer's disease, and there must be something else that might be driving someone getting this disease. So we considered that another factor might be involved is iron. And to look at this, we measured the amount of iron in people who had died and donated their brain, and according to different diagnostic categories. So here is the amount of iron in the brain of people who had died without a clinical diagnosis of Alzheimer's disease, so they were cognitively normal when they died, and they didn't have any pathology within their brain either, and that was confirmed by a pathologist. Here's another group of people who they had a clinical diagnosis of the disease, but they didn't have a pathological diagnosis of the disease, so they were misclassified as Alzheimer's disease. They had a dementia that wasn't Alzheimer's disease. Then there's a group of people who didn't have the clinical diagnosis of Alzheimer's disease, yet the pathologist said that they had the pathology equivalent to that of someone with Alzheimer's disease. So this, again, speaks to the 30% of people who have the amyloid pathology, yet not the symptoms of the disease. But they have the iron level similar to those who are controlled. And then when we looked at the people who had a pathological diagnosis of Alzheimer's disease and a clinical diagnosis of Alzheimer's disease, these are the individuals who had elevated iron within their brain. From a pathology point of view, we can't distinguish between these two groups of people. They've got the same amount of